Ever wondered how scientists identify and quantify proteins in a sample? Well, in the world of biological research, understanding the presence and quantity of specific proteins can be crucial. It can help researchers decipher how cells function, how diseases develop, and how potential treatments might work. Imagine you're a detective, and proteins are your suspects. You need to know who's present at the scene and in what numbers. That's where a vital technique called Western blot comes in. Western blot, which might sound like a cowboy's handkerchief, is a widely used method in biochemistry for protein detection. This technique allows scientists to not just identify proteins but also determine their size and how much of them exist in a sample. It's a bit like a protein identity parade, allowing scientists to pick out individual proteins from a complex mixture. Western blot technique is an essential tool in the biochemical laboratory, helping scientists to unravel the mysteries of the protein world. The first step in the Western blot technique is called gel electrophoresis. This process is a bit like a race where proteins are the runners and the gel is the track. The proteins run, or rather move, through the gel, and it's not just their speed that matters, but their size and charge as well. Imagine the proteins as runners of various heights and weights. The smaller, lighter ones can zip through the gel quickly, while the larger, heavier ones take their time. That's how we separate the proteins based on size. Now, the charge plays a part too. Proteins carry a negative charge, so when we apply an electric current, they're drawn towards the positive end of the gel. The stronger the charge, the faster they move. This way we can separate them based on charge as well. And voila! After some time, we find our proteins neatly lined up in the gel, each in its own place. Once the gel electrophoresis is complete, we have our proteins neatly separated and ready for the next step. After gel electrophoresis comes the transfer step. And this is where we take our journey from gel to membrane, a trip that our proteins must make to be accessible for further analysis. This is accomplished through a process known as electroblotting. Here's how it works. We prepare a sandwich of sorts with our gel and a specialized membrane, typically made from nitrocellulose or PVDF. This sandwich is submerged in a transfer buffer, a solution that enables the migration of proteins. When we apply an electric current, proteins from the gel begin their journey towards the membrane, driven by their inherent charge. These proteins, though invisible to the naked eye, leave their imprints on the membrane as they pass through. It's a bit like footprints in the sand, except these footprints are protein bands. The result is a mirror image of our gel, but on a membrane, providing easier access and manipulation of the proteins. With our proteins now on a membrane, we can proceed to the next step, probing. The final steps in the Western blot technique involve probing and visualization. Now, after the proteins have been transferred onto the membrane from the previous step, they're ready for probing. Here, we utilize specific antibodies that have an affinity for our target protein. These antibodies bind to the protein, essentially tagging them for us. But how do we see these tagged proteins? Well, that's where a secondary antibody comes into play. This secondary antibody, like a molecular sidekick, binds to the first. But the real trick up its sleeve is that it's usually linked to an enzyme that can produce a color or light when exposed to the right conditions. This gives us a visual marker, allowing us to see where our target protein is on the membrane. By detecting the light or color, we can not only locate our protein but also make an estimate of its quantity. And there you have it, the Western blot technique. A simple yet powerful tool for identifying and quantifying proteins. So, to recap, the Western blot technique involves three main steps. First, we have gel electrophoresis, where proteins are separated based on their size. Then comes the transfer, where our proteins move from the gel onto a membrane. Finally, in the probing and visualization step, the proteins are detected using specific antibodies and visualized. This powerful method allows for the precise detection and analysis of proteins. With this technique, scientists can confidently identify and quantify proteins, pushing the boundaries of our understanding of biology.